Alright guys, I want to talk to you guys about the Calvin cycle. So the Calvin cycle is the light independent reaction. So before we were talking about the light dependent reaction, or it's called the light reaction sometimes, that involves all the photosystems. And so now we have created the ATP and the NADPH, and the NADPH is that electron carrier that is carrying those high energy electrons. And we're going to take all that energy from the NADPH and the ATP, and we're going to put it to use in the Calvin cycle. Now, if you remember, the light-dependent reaction occurred on the thylakoid membrane. The Calvin cycle is going to occur in the stroma, so that fluid that surrounds all the thylakoids inside of the chloroplast. That's where we're going to be talking about with the Calvin cycle here. So we're going to reduce carbon dioxide to sugar. We're going to talk about what reducing means here in a moment. So there are four main steps, and the steps of those are carbon fixation, reduction, release of the G3P molecule, and regeneration of the RuBP molecule. So all I want you to know about these molecules right now are just their names. All right? We're not going to get, or their shorthand names here. We're not going to get too detailed on what they're made of and everything, just that they're being used here in this cycle. So we're going to use the carbon from carbon dioxide and the electrons from NADPH and the energy from the ATP, and we're going to do all that, throw into a cycle, the Calvin cycle, and create G3P. Now G3P is like a building block molecule that can be used to make a whole bunch of different sugars. The main one we always talk about is glucose, and that's what we're going to focus on here, because after the glucose is made, then we're going to talk about cellular respiration during another class and how we can break that glucose back down and release a lot of energy from that. So here's our overall reaction, just to give us a look at where we've been and where we're going. So again, we were talking about the light reaction, and we made the ATP, and we captured those two high energy electrons, and we're sending them to the Calvin cycle. The carbon dioxide comes in as well, and all of this is going to create some sugar. And we're going to also create the ADP again and NADP plus again, so they can be recycled back in the light reaction and then come back to the Calvin cycle. A lot of recycling going on here. All right, so we have four main steps here. We have the carbon fixation step. We have the reduction, the release of one molecule of G3P, and the regeneration of RuBP. So let's go ahead and take a look at our there's a big blown up picture there of our process here. So let's take a look at the first step here, carbon fixation. Now I got some lines drawn here. We'll take a look at those lines here in a moment. But as you can see in step one, carbon fixation, the enzyme Ribisco attaches carbon dioxide to RuBP. So enzymes are used to build and to break down. And in this case, we're building up, and we're going to take the carbon dioxide, and we're going to take the RuBP, and Ribisco is going to put them together and create it's going to rearrange the carbons. Each one of these black dots here are carbons. It's going to rearrange those carbons and the other elements that make up this molecule and create this 3-carbon compound. So let's take a look here. We have 1, 2, 3, 4, 5 carbons. 5 times 3 is 15. If we add another 3 carbons, 15 plus 3 is 18. Well, if we take a look here, we have 3 carbons here, so 3 times 6 is 18. So as you can see, we're just rearranging the molecules here. So carbon fixation is we're taking carbon and we're fixing it onto another molecule there. So since we have added that 1 carbon from the carbon dioxide to the RuBP, we now have a 6-carbon compound. This 6-carbon compound is very unstable, and so it will split into 2 3-carbon compounds. All right. So for every get this little slideshow thing to get out of the way. Come on, get out of the way. So for every carbon dioxide that enters, 2 3-carbon compounds are made. So that's where we're getting this 6 from here. Okay. So we had 3 carbons come in, and so we can make a total of 6. 3 times 2 is 6 of these here. All right. So since those 3 came in and attached to the RuBP, we can rearrange them and make 6 3-carbon compounds here. Now, we're going to take the ATP, the energy from the ATP, and the energy from those hydrogen electrons of the NADPH, and we're going to use that energy to, again, rearrange this molecule here into G3P. Now, it doesn't look like it's changed, but it actually has. Again, we're just tracking the carbons. It's a very simplistic view. There's still 3 carbons here. There's still 3 carbons here, but there has been a rearrangement of the chemical bonds and the elements in there. So this is called reduction. And so what I need to remember here is oil rig. Oil. Oxidation is losing, and reduction is rig. Reduction is gaining. What are we losing and gaining? Electrons. How? Well, all atoms have electrons that orbit the nucleus. So when we take an atom from one molecule and we take it out and we attach it to another molecule to make a different compound, what we've done there is we've transferred electrons. One of them is gaining the electron, so it's being reduced. It kind of seems opposite, like we'll reduce, don't you lose? But in chemistry terms, and you'll hear this again when you take chemistry next year, reduction and oxidation, those are types of reactions. But so we're moving the electrons from one compound to another, and we do that by transferring the atoms there. So that rearrangement there is reduction. So we have now made 6 G3P molecules. From there, we're going to step 3. Step 3 is we're going to release one of those G3P molecules. And why do we do that? Again, that G3P molecule is a building block up to a sugar. And so we're going to use one of those to create a sugar. The sugar that we like to talk about a lot is glucose. And we're going to be using glucose in cellular respiration. And so this whole process here, let's focus on glucose. It can make other sugars, like cellulose, to make the cell walls. But we're just going to focus on glucose here. So we're going to take one of those, 6, and then we'll get 5 left over. Those 5 will be rearranged again with some energy of the ATP and put them back into a rearrangement of RuBP. So let's take a look here. 5 times 3 is 15. 3 times 5 is 15. Again, we're just rearranging. So we do this, and then it goes back into the cycle. Rubisco grabs those 3 and grabs 3 more carbon dioxide and does it again. All right, so this whole process is to make that glucose there. So let's just rehash here. In goes carbon dioxide with the RuBP. The Rubisco takes both of these, rearranges them into 6 3-carbon molecules. Those 6 3-carbon molecules are rearranged with the energy of the ATP and the NADPH. These we got from our light-dependent reaction. And they move into now, they get rearranged. We use the energy to rearrange these 3-carbon compounds into G3P. Out of these 6 G3Ps, one of them is sent out, and we use it to build up glucose and other compounds. Then 5 of them go back into the cycle, or stay in the cycle, really, and we use the energy from ATP, and we rearrange them again into RuBP so we can start the process again. And the plant takes in more carbon dioxide, and we keep on making more and more sugar, and we use that sugar again for other cellular components and just for energy of the cell so that it can power its cell processes. And we're going to talk about how we break down that glucose molecule when we start talking about cellular respiration. Again, here is kind of where we've been, what we've done, where everything is located. Nice little overview here. So the light comes in to the light reaction of the thylakoid membrane. Water comes in as well. From there, we go to photosystem 2, the electron transport chain, and then photosystem 1. Out of photosystem 1 comes our ATP and our NADPH, those high energy electrons, and they'll go into the Calvin cycle, which is located in the stroma, that fluid that surrounds the thylakoids. With carbon dioxide, that RuBP is rearranged into those intermediates that we talked about, and out comes G3P, one G3P, which we can turn into different sugars that we see here. And then 5 of those RuBPs continue on and are reused again in this Calvin cycle. We can see that the ATP that we use in the Calvin cycle and the electrons of the NADPH that are used up, well, now we have the NADP plus again to go back and pick up more electrons, and the ADP to go back into the thylakoid to become ATP again through that ATP synthase process in the light-dependent reaction. So it's a lot of recycling going on here. We want to make sure we understand our reactants: light, water, carbon dioxide. Those are reactants. Our products: oxygen, sugar. We also want to take a look at what our reactants and products are here as well. NADP plus and ADP are reactants here, and ATP and NADPH are products here. All right, so next time in class we'll review this together, and we'll go from there.